Okay. And Megan, why don't you try leaving so you don't have to sit here the whole time and see if I make sure that I can still be the host, which I should be. And if there's a problem, okay. I'm still recording. Yes. Okay, great. Everything seems to be working fine. Okay. Yeah, let me just chat Megan real quick and tell it weird. Yes, thank you. Okay, great. Okay, I think that we should go, and this is somewhat arbitrary. This is my directorial vision, that we should go in the order of Ray and then Kitty and then Rob. And then
Okay.
Ray, you can come back and ask me about, and then me last, about ontologies.
So, so since I'm going to have to turn off my video, do you want to do a quick say who you are first, like a 2-second,
Yeah.
who are you? And then I'll say, I'm sorry, I have to turn off my video.
Let's do that.
And then
yeah, we'll do that.
Otherwise, I'll just be freezing and I won't hear you.
Okay. Okay, let me make sure I remember Ray Berner, Kitty Emery, Rob Grolnick, because sometimes I blank on names. So let me make sure I don't like this.
Me too. Yeah.
Okay. All right, I'll do that. So I'm going to go ahead and start. Okay, hi, my name is Ramona Walls. I'm a member of the FuTRES project. So we're going to talk to you briefly today about some of the work that the scientists and the PI, who are PIs on this project, are doing with their data, combining it in FuTRES. Let me just quickly say who we've got on the call. First is Ray Berner, who's at Howard University. We have Kitty Emery, who's at the Florida Museum of Natural History and University of Florida, and Robert Grolnick, who's also at the Florida Natural History Museum at the University of Florida. Kitty's going to turn off her camera real quick. She's got some internet issues. Thanks, but she'll be talking to us in a minute. So I'm going to start by asking a question of Ray. Ray, could you please give us a little background on how you got started working with FuTRES and where you plan to take it in the future?
Sure. I'm a vertebrate paleobiologist, evolutionary biologist. I work on Neogene and Pleistocene Old World mammals. And the last 20 years, I've had focused research interest in the evolution of horses, the equidae. I started studying three-toed horses at a site called Maragay in the mid-70s. I worked there 3 years. And I was challenged to define what species are there. And I ended up determining there were 6 species belonging to 3 lineages. So I collected a lot of data from Maragay and comparative data from other sites for systematic biostratigraphic and biogeographic work I did. And in 1981, we had a workshop at the American Museum which standardized measurements that we're using. And since 1981, I've been using these standardized measurements. They've published by Eisenman et al. In 1988 and Bernor et al. 1997. And over that duration of 46 years, I've acquired over 1,000 records on fossil horses. These records include standard measurements, and they were originally geared for systematic biogeographic and paleoecological studies. I have since 2000 also acquired a large complement of digital images of these horses, which allows me to expand the measurement records into discrete variable records and records on paleodiet, mostly mesoware. Well, with FuTRES, is coincident with me starting to work on Pleistocene localities and moving from strictly three-toed horses to one-toed horses, and that's the Equus lineage. So I've been able to do this because I'm working in younger time horizons in Africa, but also I've been working with a graduate student at the University of Pisa on the origin evolution of Equus. And we were able to decipher and interpret the evolution of zebras and asses stemming back to the early Pliocene of North America, the Hagerman horse quarry. So Omar and I have acquired a lot of measurements and digital images of both extant equus, that's zebras and asses, as well as European Stenonian horses and horses from North America. We'll be joined studying the Hagerman collection, which is an extensive collection. It's in a number of institutions, but the Smithsonian has the largest collection. It has 60 large paleo cabinets filled with prepared equus fossils from the Hagerman of Idaho. We'll be joined by Helena Mikado, University of Oregon, who studies North American and South American equus. And we intend to continue acquiring both measurement data and digital images of this collection for their two theses. Omar's thesis is going to be on Eurasian and African equus relations. Helena's will be on North American and South American equus relations. Besides that, I'm working with Gina Semperbon on paleodietary data. We're going to increase our records of not only equus, but ungulate data, principally ruminant data from Africa and Asia. I'm also working with Juha Saarinen on body mass and ecology of fossil and extant equus. This is a project that I took on specifically because of FuTRES. FuTRES is very interested in body mass estimates. And the last thing that I want to talk about is a paper I'm doing with Christine Janis on crown height. Crown height has been used by the NOW database for predicting current and fossil climate environments. So I'll be working with her on a project on asynchronicity in crown height of North American and Eurasian and African equus. We know from the paleontological record that crown height increased in North American lineages several million years before they did in Eurasia and Africa. And this is reflective of paleoclimate change occurring towards more open country savanna faunas in North America prior to them doing so in Eurasia and Africa. So that is an ongoing project.
Wow, you've got a lot going on, and it's great to hear how it all gets tied together using FuTRES, and especially the way that we can connect the paleo data that you're collecting with modern data. On that note, we're going to switch to a slightly more modern viewpoint, which is Kitty Emery, who's working on zooarchaeological data. And Kitty, would you like to give us a little bit of background on your work with FuTRES?
Sure, I would love to. And I'll turn on my video once again just to say hello. And then because I'm sure I will be freezing, I'm going to turn it off again.
Okay.
So I'm a zooarchaeologist, and I work primarily in the Circum-Caribbean, mostly in the Maya area and in the Southeast USA. I'm a curator of environmental archaeology at the Florida Museum, and our collection has an enormous set of not just the specimens from the last 10,000 years in the Southeast USA and Circum-Caribbean, but also data on traits that at this point we're using metric traits. So my primary research interest has always been understanding human impact on ancient environments, and I primarily track that through zooarchaeology, but I also use other environmental archaeologies and collaborative work in some of my projects. So one of the primary puzzles is always how to separate the human impact from the myriad of other impacts that can cause changes in populations and body shapes and everything else. And so when I try to use things like foraging ecology to look for resource depressions, I have to understand that my data isn't necessarily showing what I think it's going to show. So climate change is a big driver in both the Maya area and Florida area history. So that's something that I really need to be able to section out of the puzzle. So one thing that's really important is being able to get a full historical perspective. And for that, I need to be able to mesh paleontological data with modern data to see how animals have changed over the full range of history, pre and post, well, not post, but pre-humans and up to modern day. And FuTRES provides a huge opportunity to do that. It provides the opportunity to bridge those disciplines, which for me is essential, but also to compile huge data sets that bridge it in a way that make the data comparable. And that is a big issue whenever I'm trying to compare my zooarchaeological data to modern data, as I'll talk about in a minute. So my main project using FuTRES data right now is that I'm collecting and compiling, as I said, linear metric data from the zooarchaeological collections in the museum. And I'm collecting data on both primary prey animals and non-prey animals that are usually intrusives in the archaeological deposits, so that I can compare chronological trends in the changing body mass. Then I correlate those data with archaeological evidence of settlement change, environmental archaeology evidence of landscape changes, and climatological data. And that allows me then to test whether the prey body changes are happening at different tempos or in different ways than the non-prey body sizes, which would allow me, hopefully, to isolate human impact as opposed to all the other things that happen. So getting back to the bridging issue, paleontological data is great because we tend to use the same skeletal allometries, although we often use different baseline data, which collaborating here with Ray and other paleontologists has been really interesting to see that the modeling formulae and the constants that we're using are often coming from quite different places, which has been interesting for me. So that's something that is really valuable to understand as we're going through this. But my bigger issue is merging my data with that from people like Rob, who neontologists, because their body mass data comes from live animals or carcasses. And when they use allometric formulae to fill in blanks, they're using things like hind foot length. But we use skeletal allometries. And our skeletal allometries, there's a big gap between understanding what the skeleton and the skeletal parts can be used for in terms of metrics and our allometries, and something like hind foot length or body mass to body length or something like that. And looking at Rob's research and the research of other neontologists, there's a lot of assumptions that go into those basic allometries for modern carcasses. So my other interest is more a methodological one, bridging the skeletal to carcass metrics, so that we can actually have that specimen level data with the fine-grained spatial coverage that I'm going to need if I'm really going to separate human impact from climate or landscape change or something else in specific areas. So what I have is fairly broad understanding of how much does a deer weigh based on its toe bone. But I need to understand how much it would weigh in one area versus another area, and one set of conditions versus another set of conditions. And that kind of information can come from the huge biodiversity databases. But I need to be able to link to it using skeletal data. So that's my primary two research projects right now. However, I'm very excited about the future when we start to open up to more taxa, because one of my other research projects is looking at turkey domestication over the entire process, from wild through heritage turkeys, so essentially modern heritage turkeys in Mesoamerica, the homeland of turkey domestication. So when we start to include birds, I have enormous metric data sets that I'm really looking forward to meshing with the paleontological information, the modern information. So there's a lot of potential moving forward. And that's me.
Very exciting. Thanks, Kitty. And yeah, it's been a real eye opener for me to learn the different ways that people are measuring, doing body mass allometries between paleontology, zooarchaeology, and modern data. And on that note, we will switch over to Rob, who is doing modern data and some other things. And Rob, why don't you give us an overview of what you've been working on with FuTRES?
Sure, Ramona. So I'm Rob Gorelnik. I'm a curator of biodiversity informatics at the Florida Museum, as Ramona mentioned. And I am broadly interested in macroecological and macroevolutionary pattern and process and how that relates to global change. And to power my research, I rely heavily on natural history collections. And really in the last 20 years, digitization has unlocked collections data for broader use, which has really been a sea change that has positively transformed the core of the research collections enterprise. So what was effectively once a disconnected set of resources that required enormous human effort to assemble into a coherent picture, that really has now become a networked, digitally sort of activity that has accelerated advances in the sciences. But with all this digitization, there are still gaps in what we have available and what we can use for answering questions about how traits have changed over time and space. And so in many cases, natural history collections often record trait data. Those have been reported from the enormous efforts of field biologists to basically capture those data in the field when they've collected specimens. And so they often report, body mass and body length. but those really important characteristics of organisms are often then, when digitized and made available, not in standardized or harmonized forms. And so they're not really easy to use well by others. So back in 2016, I started on an adventure to unlock those trait data. And really at the start of that, I was interested in this question about whether we could begin to assemble spatial and temporal information about traits. So we could ask, really, really super simple questions about ecogeography. And there's been classic work going back hundreds of years to look at questions about relationships between, say, climate and body size. And those are often related to these ecogeographical rules like Bergmann's rule. And those have been really important empirical and theoretical constructs to understand why organisms look different and have different body masses and body characteristics in relationship to drivers that might have been operating in terms of selection over time and space. But we haven't really ever had the chance to assemble this huge view of what happens across thousands and thousands of species. And that was really the impetus for wanting to go and begin to standardize these reports of body metrics that we could gather from specimens, from individuals. And one thing I'll mention about this is that there's a lot of body mass data out there, but often it's at the species level and not at the specimen or individual level. And that really limits the kind of inferences and the kind of power that you can use to understand how to scale between these sort of interest-specific aspects of variation all the way out to these intraspecific and often these clade level aspects of variation. But having data reported at the individual level allows us to begin to kind of connect the dots between the interspecific and the interspecific variation. So we spent a lot of effort to harmonize and standardize those body mass metrics, but we also recognized that there were lots of other body traits that we weren't capturing in that first pass. So although we were able to assemble, harmonize, and standardize, almost 1 million or more than 1 million observations of mass and length from available collections data that were already digitally available, we knew that we were missing, information about individual elements that are often really important for linking back to these allometries that are of interest to people in the archaeological and paleontological communities. And we also knew that we had other kinds of traits that we could capture that were of interest, like reproductive traits. So those were thoughts that we had about what we could do next. And happily, we begin to see a path for sharing those data more broadly and fruitfully and in an interconnected way with FuTRES. But one of the things I'll mention before I come to FuTRES is that just to sort of prove that these kinds of data could be accumulated and useful, the very first thing we did was we started looking at large-scale questions related to Bergmann's rule and trying to understand if Bergmann's rule actually applies for mammals especially and also for birds across thousands of species. And what really was fascinating from the initial work was that we didn't find a strong Bergmann's relationship or Bergmann's-Klein, which really began to get us thinking about what's going on if it's not generally true that Bergmann's rule holds for lots of species. How do we begin to understand more about what does drive body size and body size change? And may it be landscape effects, and maybe that body mass is driven by other factors that are happening in the present, like urbanization or other kinds of drivers. And so what started as a sort of a beginning exploration of how to get the data assembled became this larger exploration of how we can begin to sort of piece together stories about the drivers of body size, differences across species and within species. And so that's been really pushed forward my research agenda a fair amount. But FuTRES has kind of amplified and helped me see, even the broadest perspective of thinking, especially about temporal contexts that extend beyond the ontological sort of modernish timeframes, say like the last 100 years of collecting. What's really exciting to me is with the integration of interdisciplinary expertise in this project and the strong focus on informatics to help us standardize and report how we know what we know about traits more effectively. We're really at the precipice of beginning to be able to link across not just the, the temporal and spatial scales, but also the scales almost in the sense of the the structural elements that we're comparing. So trying to link between, say, a metacarpal metatarsal to a whole forearm up to the whole body length or body mass of an individual. Each of those components and those scalings and how we can understand those, we've really had, basically pieces of that puzzle to assemble. We haven't had the whole enchilada to kind of pull together. And for me, FuTRES is a super exciting project because it begins to show the power of integrating these data in a single framework where we can access, basically the core information about what we've measured, how we've measured it, and then begin to link up and link out, across the sort of the spatial organization of organisms to the space and time issues that drive, why we have this enormous variation in body size and body conditions across a group like mammals or any group of animals at all. So I'm excited to move forward with FuTRES and see where we're going. I've also just really appreciated the interdisciplinarity of the project.
Mm-hmm. And I'm just excited to see the entire enchilada being made, because that's really what drives me about, pretty much everything. It's Mexican food. So no, but it's great to hear the different perspectives, the different kinds of research. And I hope that people who are listening get a sense of the real broad range of questions that can be asked with the kinds of data that we're collecting in FuTRES, and how even though each of these individual researchers is doing very complicated, really large, they're already gathering really large data sets, doing lots of work to get those cleaned up and standardized internally. But then we're adding a whole nother layer to bring all three of those data sets, plus many more that we're not talking about here, together into an even bigger, more complicated picture. Are there any questions that any of you would like to ask? Yes. Kitty.
I would actually like to ask the interviewer to talk about ontology and talk about how, like, the function of bringing this all together, because that's a super important component of this.
Absolutely. I'm going to give a brief answer to that. We will have, during the actual live webinar, we're going to have a great presentation by Brian Stuckey from the University of Florida, who's going to go into this in a lot more detail. But I will just say that my role as an ontologist and as a data manager is then to sort of look at what each of you are doing. As I mentioned, you've all done great work to clean up, standardize, and harmonize your data internally. But then my job is to try to bring all of those different data sets together. And the real challenges, there are two main challenges. One is sort of the basic data cleaning that I think many of you are already familiar with. Even though data sets have been cleaned up, we go through them, we find more issues. I was just talking with Ray before the call about there might be a question, a comma versus a period in a decimal point. How do you find these things? Fortunately, I have a wonderful postdoctoral researcher, Megan Balk, who has been spearheading those data cleanup efforts and has done a ton of work there, along with various grad students who have been working on this. But then once we get the data all cleaned up, we still need to make sure that the different data sets work together, and that's where the ontology comes in. The ontology really provides two important pieces of harmonization. The one that most people would recognize and the one that matters most to the scientists is really defining the different traits in a way that makes sure that they're compatible with one another. So I have a background in plant biology. I had no idea that there were so many different ways that femurs could be shaped. So the little bumps on the ends of the humerus and the femurs, how they come together across deer, horses, primates, all the different animals. Apparently, depending on how you walk, your femur can be very differently shaped. And so when you say something like femur length, if you're measuring the length of a femur, that's going to be really different in a horse or a cow or a deer or a rodent, for example. And so in the ontology, we're able to preserve the very specific, sorry, my screen just went away. We're able to preserve the very specific definitions that apply across the different taxa, but then still make generalized terms that bring them all together. So a person who wants to measure a femur length that goes to the tip of some little knob can get just that specific data. But if they want all femur lengths across all animals, they can grab those as well. The other really important role that the ontology plays in this project is a sort of generalized data model. It allows us to link together data from the location where the specimen was collected, to maintain that link to individual specimens, to talk about the environment at the location where it was collected, to gather all of that really important archaeological and paleontological and zooarchaeological metadata that Ray and Kitty are collecting. And the ontology provides a really nice data model that's very generalizable across essentially any data set that we can throw into it. So that's what I'm doing with the ontology. Any other questions or comments from all of you?
I have a comment. One of the most useful aspects of me being involved in the FuTRES project is getting my data set in shape to publish. Mm-hmm. And when you're acquiring data and entering it into a spreadsheet or a database, there's a tendency to add additional things like an E for estimate or comments like not measured and things like that. And it takes several sweeps. I think I have 14 versions and 22 different saves on my data set over the last year and a half, and it finally got cleaned up last week. So this has been an extremely useful thing to do. I mean, normally you have a data set and then you take a subset of that for statistical analysis and you clean that set so it'll run through the statistical package you're using, but you don't go back to the original data set and clean up behind you. Mm-hmm. Another thing is you change the taxonomic names from a general name to a specific name in a scientific paper where you have to go back and re-enter that new data in the data set. So I've taken a year and a half working on this steadily, and it's been very useful to have the FuTRES framework and program to compel me to do that.
That's great. Yeah. And I think that working with others and getting ready to publish your data is a great motivation for people to do that kind of cleaning. And we're lucky that we have the postdocs and students who have been able to write great scripts to help us clean up some of this data and to standardize it and to make templates that other scientists can then use to get their data into, to make it easier for new researchers to bring their data into FuTRES. We hope that we'll have R packages, et cetera, that will make it easier for people to clean up their own data. On that note, unless there's any more urgent comments, I think we're going to wrap up, and hopefully we will be talking to you all in person during the workshop very soon. So thanks so much, everybody.
Thank you, Ramona.
Bye-bye.